Let's get a little bit bigger now and take a look at the cell and some of the subcellular structures inside that we call organelles. Now, before we do that, you need to realize that there's around about 30 trillion cells in our body, and not all those cells are the same. Some cells are really small, like a red blood cell, sometimes just referred to as an RBC, and these red blood cells are only around about 10 micrometers in diameter. Now that's very tiny. If you think about when it goes 1 meter and then divide that by 1,000 and you've got 1 millimeter, and then divide that by 1,000 and you've got 1 micrometer. And so 1,000th of a millimeter is a micrometer. So it's only 10 of those. That's the size of a red blood cell. So tiny. You can have a sperm cell, which isn't too much bigger. The head of a sperm cell is actually smaller than the red blood cell, but the tail can be about 50 micrometers. And then you've got really big cells like an ovum, which is the egg cell, the ovum, and this is a lot bigger. So this is around about 100 micrometers in size. And you can even have some cells lengthwise like neurons, and they can be 1 meter in length. So cells come in all different shapes and sizes is the point I'm trying to get across. But what I want to talk about now is that the fact that all cells in the body share some similarities. And so I've got a cell here which has a couple of important points we need to be aware of. Let's first start from the outside. And we already know that a cell is surrounded by something called a phospholipid bilayer. So if you have a look, if we were to take the magnifying glass and just focus here, what you would find is that you're going to have 2 layers like this called the phospholipid bilayer. And it goes all the way around. And remember, it's made up of phospholipids, and the phospholipids have a head on it, which is made up of phosphate. And that phosphate loves water, and it's made up of a fatty acid tail. Actually, 2 fatty acids. And that hates water. And so it arranges itself where the things that hate water are away from the water. They're just basically articulating with each other. And then the phosphate head is exposed to the water. So the phosphate head is going to be exposed to the water inside the cell. So it's going to be here. And it's going to be exposed to the water environment or aqueous environment outside the cell. And so this is going to be the phospholipid bilayer. This is important because, remember, it's semi-permeable. It's semi-permeable. So that means it lets some things in and other things it doesn't. Do you remember what it does and doesn't let in? Remember, if it's large or charged, it's not getting in, right? So that means ions can't get in, for example. Gases can get in. Large molecules like glucose can't just move through. They need channels to move through, okay? All right, let's go deep inside the cell. And we're going to take a look at, first of all, the nucleus. So we've got the nucleus of the cell right here, and it's cut open. And you can actually see a smaller nucleus inside. This is called the nucleolus. And again, this is called the nucleus. Now, both the nucleus and the nucleolus house DNA in the form of chromosomes. So DNA is a long stretch of sugars, nucleotides, right? A's, T's, G's, and C's. And they can be wrapped up really tightly, because if I were to take one cell from your body, pull the DNA out of the nucleus of that cell and hold it so that it's one end in my hand and the other end on the floor, it would be about 6 foot long. So that's in every cell, 6 foot of DNA in every cell. So it needs to be tightly packed. And that's when it's in the form of chromosomes. That's tightly packed DNA. And you're probably thinking, well, why is there a nucleolus and a nucleus? It can't house the same things. Well, I will get to the nucleolus in a second. But the DNA in the form of chromosomes are housed in the nucleus itself. If we move a little bit out, you can see that attached to the nucleus is this structure here. Now, this structure is called the rough endoplasmic reticulum. The rough endoplasmic reticulum. Good name. Rough endoplasmic reticulum, sometimes just referred to as the rough ER. Now, it's not shown in this picture, so we're going to draw it up. It's called the rough ER because it's studded by all these little dots. When you look at it under a microscope, there's all these dots on it. What these dots are ribosomes. So let's label that. These are ribosomes. And you can have the ribosomes studying the rough ER, or you can have free-floating ribosomes like these ones here. What do ribosomes do? Well, they take the RNA, so think about this, right? Inside the nucleus, you have DNA in the form of chromosomes. These chromosomes need to create proteins. So some of that DNA needs to be read to be turned into proteins. And the way we do it is that in the nucleus, DNA is translated into RNA, and then that RNA is turned into amino acids. Now, the DNA to RNA, this whole process happens in the nucleus. But RNA to amino acids, this happens in ribosomes. And so it can happen. You're looking at it and you're going, but there's two types. There's ribosomes attached to the rough ER and there's free-floating ribosomes. So here's the difference. The free-floating ribosomes, like these ones here, they create amino acids. So they translate this RNA to amino acids to remain inside the cell. Inside the cell, right? The ribosomes that are studying the rough ER, they turn RNA into amino acids that are destined for export out of the cell. So let's stick to this process, right? Let's stick to the ribosomes on the rough ER. The nucleus has spat out some RNA, given it to the rough ER. The ribosomes turn it into amino acids, and then they pass it off. They pass it all off to this structure here. Now, this structure here is called the Golgi apparatus. Golgi apparatus. And what the Golgi apparatus does is it packages up the proteins that the rough ER has just made, slaps a stamp and sticker on it to say where it needs to go, and it starts to export it. So it creates this vesicle. I'm going to make it a better color. So it creates this vesicle, which is just like a very small cell, right? It's membrane bound, and it's going to have the protein or proteins inside, and it starts to send it for export. It fuses with the wall here, and then spits out those proteins for export, called exocytosis. Exo meaning exit, and cytosis meaning cell. That's what the Golgi apparatus does. So you could think about, right? You could think about the nucleus being the library, contains all the information in the form of DNA. Then you've got the factory, which is the rough endoplasmic reticulum. They build stuff. What they're building are proteins. They can send those proteins off to the Golgi apparatus, which is like the post office. Packages it up, slaps a sticker on it, tells it where to go. Now, what we call this is post-translational modification, because it's already translated from RNA to amino acids. That's translation. So when it modifies it, it's called post-translational modification. That's what the Golgi apparatus does, and then sends it away. There's actually another type of endoplasmic reticulum, which isn't shown here, but I'm going to draw it up. So there's another endoplasmic reticulum which looks very similar to this one, but it doesn't have RNA, so it's not studded. And this is called the smooth endoplasmic reticulum. Smooth ER, smooth endoplasmic reticulum, because it doesn't have the ribosomes. So its job is not to synthesize proteins, but its job is to synthesize fats. It also detoxifies. So if you have any drugs that need to be broken down and detoxified so they're not damaging, that's what the Smoothie R does. Now remember, the liver plays a huge role in detoxification. So looking into the cells of the liver called hepatocytes, if you look inside, they've got huge amounts of Smoothie R because they play an important role in both synthesizing fats but also detoxifying substances. Now, I haven't told you what the nucleolus does. The nucleolus produces ribosomes. And I've already told you what the ribosomes do. Beautiful. Now, last couple of things is the mitochondria. Got the mitochondria right here. Now, the mitochondria, you probably always hear, is the powerhouse of the cell. What does that mean? It produces ATP. That is energy, synonymous with energy in the human body. So it takes glucose, so a simple sugar, and it takes oxygen and produces huge amounts of ATP for energy. Now, mitochondria, evolutionarily speaking, millions and millions of years ago, I should say billions of years ago, was its own separate organism, akin to a bacteria, and it fused with an early organism, what I should say is an early family member of ours. And then we became reliant on that mitochondria to produce our energy. Brilliant. It actually has its own DNA. So very separate to our cells, where the DNA is sitting in this nucleus, mitochondria has its own DNA. Now, there's going to be other things floating in the cell, right? Even though 75% of it's going to be water or fluid, the second most abundant thing is going to be proteins. And it's not just proteins that are made from these ribosomes for function. So some proteins carry stuff. They're used in enzymes. They're going to be receptors. They're going to do a whole bunch of stuff. Some proteins are structural. And so you can have proteins that simply just act like scaffolding for the cells. They keep it stable. They keep it strong. One type is called microtubules. Now, the last two things I want to talk about are these little things here. These little things, there's going to be two different types. There's going to be peroxisomes and lysosomes. What they do is they break things down, basically. Lysosomes will break things down by giving things water, right? So we know that if you want to take an amino acid and merge it with another amino acid to build a peptide, you need to take water away. But if you give it water, it splits apart. And that's what lysosomes do. They split things apart using water. Peroxisomes do a similar thing, but they split things apart using oxygen and reactive forms of oxygen like hydrogen peroxide. So this is a quick run through of what's happening in a human cell.